We are the Crop Growth and Environment Group. For our experiment, we were interested in the impact of monocrop versus intercrop treatments of corn, bean, and squash. This experiment looked at the impact of biodiversity on crop growth and environment. Temporal biodiversity was observed in the form of cover cropping, and spatial biodiversity was observed in the form of intercropping. However, for this project, our group focused only on spatial biodiversity, and we conducted all of our measurements on the plot that did not have a cover crop.
We based the research of our project around two main questions. The first being, how does a specific cropping system change the environment? We worked on answering this question by measuring relative humidity, air temperature, soil temperature, and light. The second question was, how does a specific cropping system change crop growth? In order to answer this question, we measured plant nutrition using chlorophyll content as a proxy, plant height, above ground biomass over time, and the harvestable biomass at the end of the experiment.
For chlorophyll concentration index, we hypothesized that an intercropping system would change the light availability to the crops in the shade, leading to the conclusion that the crops in intercropping plots would have lower chlorophyll concentrations than those in the monoculture plots. Using an Apogee chlorophyll concentration meter, we randomly selected 5 plants per monocropping plot and 15 plants from the intercropping plots to get an average concentration from each plot.
Digital soil thermometers measured soil temperature in Fahrenheit directly next to the plant. Sampling locations were randomly selected in the center bed, with beds on either side to act as a buffer. 3 points were taken in the monocrop treatment, 6 points were taken in each intercrop treatment, 3 for the squash, and 3 for the corn and bean.
For measuring crop height, biomass, and harvestable yield, we randomly selected 1 crop from each fallow replicate. Comparing intercrop to monocrop and looking at height, biomass, harvestable yield for corn, beans, and squash. Utilizing random selection for each sample, we measured height using a tape measure in centimeters. Next, the fresh biomass was cut at the base of the plant and harvested in paper bags, which were left to be dried in the dehydrator for the next week. All dried biomasses were then recorded in grams. Finally, in the last 2 weeks of the experiment, we weighed all harvestable biomass, which was doable for only corn and squash at the time.
For measuring evapotranspiration, we measured air temperature and relative humidity. We sampled each variable at midpoint locations for all fallow plots. Measurements were taken every 2 weeks using a digital psychrometer. The air temperature and relative humidity was assumed to change in accordance with developing crop growth sizes and environmental conditions. Therefore, the measurements were taken at varying height intervals of 6 inches, ranging from 0 for the first week and 24 by the last week. Since relative humidity is not a precise measurement for humidity, it was decided to calculate and use vapor pressure deficits to provide accurate results for measuring evapotranspiration that occurred during the trial.
Light was also measured every 2 weeks using a digital light meter, but the reading from the first week was omitted due to excess fluctuations caused by changes in cloud cover that day. We randomly selected 2 different areas from which to take light readings at each intercrop and monocrop replicate in order to measure differences in available light due to competition within the intercrop and monocrop replicates. We took readings from both the base and the top of each species of plant at each of the 2 randomly selected areas. To better represent the competition amongst plants for available light, readings were taken from the northern side of the plant in order to accentuate the effect that shading would have.
After collecting data on the chlorophyll concentrations in both the monocropping and the intercropping plots, it appeared that the overall trend showed slightly higher concentrations of chlorophyll in the intercropping plots. However, after analyzing our data, we found that the difference between the 2 plots resulted in no actual statistical difference.
Soil temperature was not significantly different from week to week or between monocrop and intercrop treatments. The highest average temperature recorded per crop was in monocrop corn at 72.5 degrees Fahrenheit, and the lowest average temperature per crop was monocrop squash at 70.7 degrees Fahrenheit.
So, in terms of the results from the measuring of height and crop biomass in intercrop and monocrop systems, we can take away a number of findings. Firstly, in terms of overall crop height, neither monocropping or intercropping systems definitively had crops that are taller in height across the board, with squash being significantly higher in B monocrop, beans being significantly higher in B intercrop, and corn being about the same height in both farming systems. Secondly, in terms of our measurement of crop biomass, a clearer trend can be observed in our findings. The squash and corn dry weight averages were remarkably higher than the intercrop measurements, with the monocrop squash being about 10 grams heavier on average than the intercrop squash. The beans in the measurements weighed roughly the same for both farming systems. So from these findings, one can say with some conviction that a monocrop farming system tends to yield higher overall biomass and dry weight of the crops grown in this system.
For the results of the observations for relative humidity, air temperature, and vapor pressure deficit, our findings show that each variable were indeed influenced by crop growth and environmental conditions. However, we observed just a small difference for each variable between monocrop conditions and intercrop conditions. Yet we did find that vapor pressure deficit to be greater overall with the monocrop compared with the intercrop, although the intercrop was higher than the monocrop corn. From the averages we found for each variable at the end of this experiment, the most significant finding is the difference of conditions at ground level compared with upper regions of crop growth. We found that at ground level, all crops had higher humidity and temperatures and tended to shift at upper regions of the crop canopy. For relative humidity, we found that ground level measurements of 45.7 for intercrop decreased to 32.37 at 24 inches, whereas for monocrops, averages of the three ranged from approximately 45.8 at ground level and decreased to 28.6 at 24 inches. The vapor pressure deficit also followed this trend. This variable also increased in values that ranged for both intercrops and monocrops. The intercrop ranged from 1.58 at ground level to 1.6 at 24 inches. The monocrop was 1.69 at ground level and 1.71 at 24 inches. A similar pattern followed for air temperature, which also changed with crop height. For the intercrops, the average ground level temperature was 23.7 and cooled to 20.2 at 24 inches. And for the monocrops, the average of the three at ground level was 24.5 degrees and cooled down to 20.6 at 24 inches.
Fractional PAR computes what fraction of the incident PAR, which is the light reaching the canopy top, is intercepted. It is abbreviated FI-PAR. It indicates the portion of PAR used by plants. From the first set of measurements, the results showed that the fraction of intercepted PAR was higher in intercrop corn and intercrop beans than in monocrop beans and corn. For squash, the difference was not significant, although the FI-PAR values was lower in intercrop squash than in monocrop squash. On 517, FI-PAR of intercrop bean was significantly lower than that of the monocrop bean. Except for this one, there were no significant differences of FI-PAR values between treatments or crops for the second and third measurements.
Understanding how monoculture versus intercrop systems affect crop environment and yield is important in implementing alternative agriculture on a wide scale. More biodiverse systems and their associated ecosystem services can provide agricultural systems with biological sources of required nutrients, storing carbon and nitrogen, providing efficient in-ground water storage, and much more. Our research provides crucial insight in proper implementation and management of on-farm practices that are more sustainable than conventional agriculture and preserve soil and agriculture for future generations to come.
Our advice for the next class: keep your data organized, print out data sheets and the map of the experiment, which you will need every time you take data. Figure out your hypothesis early and start taking pictures and video early. You will have more time during class to take measurements than you think you will. So you can definitely measure both cover crop and non-cover crop treatments, and measure both soil temp and soil moisture. You should also measure both fresh weight and dry weight for plants so that you can calculate harvest index. Although specializing data collection is efficient for fieldwork, it creates a disconnect within the group.